So in this experiment, we're given a bunch of gDNA and a particular restriction enzyme called allele, and um, we are told that this enzyme recognizes a specific sequence, AGCT. And what this means is every time the restriction enzyme comes across the AGCT sequence within the gDNA, it's going to cleave it. And this particular enzyme is actually it actually makes blunt ends, so it cuts the sequence right in the middle like that. And this AGCT sequence is actually really common in this allele family, which is a family of repeated DNA sequences, which is actually a sign essentially. So this is from the E module, but signs are small interspaced nuclear elements, and they're basically short-ish regions of there's basically like small sequences of DNA that are repeated throughout the genome in different places, hence interspaced. And the allele family is actually the largest family that makes up these signs, and they contribute to 11% of the genome. And you can actually subdivide this really into three types. So you've got the J type, and these are the oldest sequences. Then you've got the S type, which are middle age, and then the Y type, which are the youngest sequences. So we know that these allele repeated sequences will have a lot of AGCT. Okay, so that means that this restriction enzyme, whenever it comes across an allele sequence, it will cleave it essentially because it will have this AGCT sequence. And so here you can see on the left-hand side, we're given a bunch of the names of a load of different sequences within this allele family. So you can see they all have allele, and then they have J, S, or Y, depending on how old they are. And then you've got other letters and numbers we don't care about. But the way you read this is that each sequence is different, right? That's why they're different names. So if you wanted to look at the J, allele J sequence, this is the first base here, and then you would read all the way along here until you get to 100, and then you have to come down here again. Look, here's the allele J, base 101, all the way over to 200, and then the same here for 201. And we know that, or well, we presume that these sequences will have this AGCT somewhere. Otherwise, why would they give us this information? So the next step is to actually find out where these sequences are. And it turns out that they're here. And essentially, wherever there's a red rectangle, we have the AGCT sequence. And that means that the restriction enzyme is going to cleave here. So if I just focus in on this red rectangle here, which sequence does this relate to? It's the allele SC sequence. So if you imagine you're a restriction enzyme, you're going along the human genome, as you do, and you stumble across this allele SC gene. You're going to start at base 1, and you're going to move all the way along, right? You're not going to find anything. You're going to come to base 101, move all the way along, and suddenly you get this cleavage sequence. And now you've been training for this your whole life. It's what you're made to do. You know, you're going to cleave here, right? This is the sequence you recognize. So you're going to cleave here at this point in the sequence, and then you're going to continue moving along until you get to here, and you've got another sequence. Now you're going to cleave again. And what you've created is a fragment of DNA, right? From here to here. And so the question is, how long is that fragment? It turns out it's around 32 bases long, okay? And this is the 136th base I had to just mind-numbingly count from here to here. And then this is the 168th base as well. But this isn't just one DNA fragment, right? These are highly repetitive. All of these allele sequences are found, you know, countless times through the genome. So it's not just one fragment we're making tons of these DNA fragments that are 32 bases long. And that's what we see on here. So this is actually the first band, the 30 bases long. And yeah, so if we go ahead and do it for 50 bases, which is the next fragment here, you can see you've got the next band here. What happens if we take another sequence? So let's look at this ALU-YA8 sequence down here. If you're a restriction enzyme again, you're going to start here, go all the way along, nothing happening. Here, go all the way along, nothing happening again. Get to here, and you can cleave, right? Because you recognize the sequence. Then you're going to continue moving along until you get to here, and you've got another sequence. So you're going to cleave here as well. Now you've got another DNA fragment you've created, and this one goes from 168 to 218, and this is around 50 bases long. But this time, right, if you look at the first example, there's only one sequence here that has an AGCT sequence here, right? So essentially, we only have one sequence that's like making the 32 base fragment, right? Here, we've got 1, 2, 3, 4, 5 sequences, okay, that are cleaved here. And all 5 of them are also cleaved here. So now we've got 5 sequences that are cleaved to make 50 bases. The DNA fragments, that is, that are 50 bases long. And once again, these sequences are found all the way throughout the genome. They're highly repeated, so we get loads of them. And that makes up the second band here. And so now we're left with the third band, which is 60 bases long. And the annoying thing is that biology doesn't, nothing is like exact in biology. And one of the weird things, when scientists actually did this research and looked at this restriction enzyme, they actually found that a lot of the time, it cut these sequences here as well. So this green rectangle is where all of the sequences actually get cleaved. And this is an AGC sequence, so it's not AGCT, it's just AGC. And I don't really know why that's the case. Maybe the enzyme was just too excited and it read the first 3 bases and just assumed that, I don't know. But what can happen is all of these sequences have AGCT, right? All of these sequences here. So all of them are going to get cleaved here. And all of them have AGC here. So all of them are going to get cleaved here as well, potentially. And the distance from this cleavage point to this cleavage point is around 60 bases. And because every single sequence can get cleaved here and here, we're going to have the largest number of DNA fragments, right? If every single sequence can get cleaved and make this fragment, we're going to get the most fragments overall. And then you couple that with the fact that all of these sequences are also found at many different locations, means that this 60 bases DNA fragment is actually going to be the like most prevalent, and you actually see that here. So you can see the 60 base band is actually you can basically see it's the most visible, and that's because it's got the most DNA fragments, right? And so now you know why. And for those of you that are just pedantic, can the restriction enzyme cleave here and then just ignore this and then maybe cleave here, or not there? If the enzyme can potentially cleave here, ignore this, and then cleave here as well, but this is really rare, right? And obviously in this case, you'll get a DNA fragment which is much longer, but they're much rarer as well. And you can also get some DNA fragments that actually only correspond to from this cleavage point to this cleavage point. And in this case, you know, the enzyme has basically ignored the site here, and it's also ignored all of these sites as well, and it's gone along and it's cleaved here and then cleaved here. But this is much rarer, and you can see that here. So these are actually the positions of the bases, which is why I labeled them. So you've got 168, 200, it should be 229, not 228. But yeah, you know, and so you can basically compare like how much of each one there is basically. And the 60 bases, 58 or 50 bases rather, and 30 bases, those are the ones that are most prevalent, and they're the ones that are in bold, and those are the bands that show up. And you do get some of the others as well, but they're far less prevalent as well. And so this basically is the slot. This explains what you saw earlier from the face-to-face, and this is just a table that summarizes everything as well if you want to go back and compare the positions. So yeah, I hope that's explained everything.